Something happened. Let's start with our first topic under the topic host plant. I am audible now?
That screen is not visible now.
I'm doing that thing. I think something, some error happened in mid. Where is the? Is it now visible?
Yes, ma'am.
Okay. Let's begin with the host plant resistance. Our first topic under this host plant resistance is some historical perspective. Here we will see history of host plant resistance, how it came into existence, and when we were first to identify the plant resistance as the exploitable tool for controlling our insect pests. Okay, what happened in 3rd century BC? In 3rd century BC, there was a person, Theophrastus. Okay, Theophrastus, he recorded differences. He first time recorded differences in susceptibility of plants towards the fungal pathogens. Okay, he first time recorded differences in susceptibility of plants towards the fungal pathogen, and it was the initiation of the host plant resistance, and it is considered as the first observation on HPR. Okay, then comes 1782. Another landmark is 1782. In 1782, what happened? Uh, first documented record of insect resistant variety. In 1782, there is one wheat variety known as Underhill. Underhill, it was resistant to, Underhill was resistant to Haitian fly. To Haitian fly. Underhill was the wheat variety. Okay, wheat variety. Underhill was resistant to Mytiola destructor, that is Haitian fly. And it is the first documented record of insect resistant variety. First documented record means it is in the documents, it is in the papers, it is mentioned in some research papers. Okay, it happened in year 1782. The next historical mark is 1817. 1817. In this, sorghum was reported to be resistant towards grasshopper. Sorghum was reported to be resistant towards grasshopper melanoplus species. Okay, melanoplus species. Then next landmark is 1831. In 1831, this is also a very important landmark under the history of host plant resistance. One apple variety, winter mazatine was identified to be resistant against woolly apple aphid. Against woolly apple aphid. Apple variety winter mazatine was found to be resistant against woolly apple aphid, that is Eriosoma lanigerum. These names you have to remember very well. Woolly apple aphid, the scientific name of woolly apple aphid is Eriosoma lanigerum, and winter mazatine was the apple variety identified to be resistant towards this pest. Okay, then in 1885, what happened? there was a pest named grape phylloxera. Grape phylloxera, that is Dactylosphaera. Dactylosphaera. Dactylosphaera vitifoli. Okay. In North American grape root stalks were found to be resistant towards grape phylloxera. North American grape root stalks. Okay. And this was the first time when any grafting experiment was carried out for conferring the plant resistance. Okay, these root stocks are used for the development of new seedlings, and when the new seedlings developed from these root stocks, by using these root stocks, those grape plants are found to be resistant towards grape phylloxera. Then the next and last one is the 1935. In 1935, cotton was reported to be resistant to leaf hoppers. Okay, the case of resistance development emerged. Cotton was reported to be resistant to leaf hopper impoasca. Okay, it is all about the historical perspective. Next is the concept of HPR. What is HPR? Host plant resistance. Okay, see, it is very much clear from the name, host plant resistance. Host means something like insect feeds upon plant. Okay, and this plant will act as host. So when this plant will exhibit any resistance phenomena, then the whole phenomena will be known as host plant resistance. In any way the plant will able to develop resistance towards this insect or overcome the damage caused by this insect, then the whole phenomena will be included under host plant resistance. Okay, so many scientists have given the definition of resistance. One by one, scientists have refined the definitions. Okay, definition and concept of the resistance. The first the first definition you should know is of Maxwell et al. Maxwell et al. 1972. Okay, he defined resistance as heritable characteristics possessed by the plant, which can influence the degree of damage done by the insect. Those heritable characteristics possessed by the plant which can influence the degree of damage done by the insect. Okay, this is the definition of resistance given by the Maxwell et al. Okay, here what is there is in the whole definition you can see there is no existence of environmental conditions. There is no existence of environmental conditions. So coming scientists refined the definition, and you know it well that resistance can be affected through environmental conditions too. Just for an example, you just consider a case that if a person is susceptible towards cold, okay, if a person is susceptible towards cold and he goes into a hot region, in a region where temperature is nearly about 35 to 40 degrees Celsius, will he catch cold? No, he will not be able to catch cold because of his environment. He is not exposed to that susceptible temperature, so he is not catching cold. So in resistance development, there is a significant effect of environment too. Therefore, scientists refined this definition somewhat more, and what they said that if two varieties are there, okay, two varieties are there, V1 and V2, and they are having same level of insect infestation. they are having same level of insect infestation, and they are under similar environmental condition. They are under similar environmental condition. Then the variety which produces more grain, the variety which produces more grain will be resistant variety, will be considered as resistant variety, and the variety which yields less grains will be considered as susceptible varieties. I think it would have been clear. So the existence of this similar environmental condition, we can see in the definition of Kogan. Okay, we can see in the definition of Kogan. He proposed this definition in 1994, and he said that resistance to insect is inheritable property. It is inheritable property, means it can be transmitted from one generation to another generation. Okay, resistant to insect is inheritable property that enables a plant to restrain the growth of insect population, or to recover. See, here there is one more thing, recover from injury. If the injury has been caused, then there must be some ability to recover it. Even then, if plant is able to recover the injury, even then it will be considered as the resistant plant. Okay, injury caused by population that were not restrained, Okay. Next is host plant resistance is defined as the heritable qualities. HPR is the conclusive definition, defined as the heritable qualities of a cultivar to counteract the activities of insects so as to cause minimum % reduction in yield as compared to other cultivars, of the same species under similar conditions. I'm repeating this definition. This definition is somewhat holistic because it is a conclusive definition. Host plant resistance is defined as the heritable qualities of a cultivar to counteract the activities of insects so as to cause minimum % reduction in yield as compared to other cultivars of the same species under similar conditions. Okay, next is what comes next, that what are the properties or characteristics of resistance There are 4 characteristics defined for the resistance. Okay, resistance are heritable. All resistance mechanisms are controlled by one or more genes. Okay, these are heritable. They can be passed on to the progeny, to the next generation of the plants. Then second characteristic is relative. Resistance is a relative term. Okay, I said V1 and V2. We are comparing which variety is much more resistant. Then we compared the grain yield. It gave 5 kg. With the same level of infestation of insect, it gave 5 kg yield, it gave 10 kg yield. Then we can say that V1 is somewhat more resistant towards this pest. Okay. V1 is somewhat more resistant towards this pest. Therefore, it is relative term. We are saying it in comparison to V2. Okay. And if say V3 comes, then and V3 yields 15 kg, then V3 will be the resistant variety in comparison to V1. We will always prefer V3 in comparison to V1. Okay. Therefore, resistance is always a relative term. The next one is measurable. Okay, resistance is always measurable. It can be quantitatively measurable or qualitatively measurable. Okay. Then next characteristic is it is variable. It is variable. It can be easily modified by abiotic factors or biotic factors. These four characteristics are the characteristics for assessing resistance. The first one is heritable, second one is relative, the third one is measurable, and the fourth one is variable. Okay. Next, we should move to our next slide, that is host plant selection process. Host plant selection process. See, whenever an insect wants to perceive a plant, okay, whenever insect is roaming around, okay, insect is roaming around and it is exploring all available niches it can. Okay, but how we should be wondering now how any insect reaches that particular pest field where there was no infestation, how it can come with this much easily. Just we will, whenever we will observe our fields 1, 2 or 10 days or 5 days before, 5 days earlier there were no pests, but suddenly after 5 days insect infestation is seen. How this pest came into this? Okay, so there is and establishes one, and they, so in this whole process there are some sequence. Okay, one by one there are some interactive phases in which includes different kinds of mechanisms at each and every step. Okay, the first thing in host plants, in the whole host plant selection process, the first you have to remember the sequence always. Sometimes they will frame the question regarding the sequence. They will give you options with these options shuffled up. Okay, like first option, host habitat finding, followed by recognition, followed by acceptance, followed by suitability, followed by finding. Okay, second option, they will shuffle it up in this manner and they will present it to you and they will ask you to select the right option. So you should remember this uh, whole thing step by step, and this is always the whole process of host plant selection always proceeds in these steps only. There is never any variation from these steps, from the sequence of these steps. The first one is host habitat finding. What happens? It is a general approach. Okay, in this, the insect use general mechanism. It just goes with the flow wherever it can. Like it follows anemotaxis, it follows phototaxis, it follows geotaxis, whatever is available at that time with that insect, it follows it according to its preferences, like humidity preference, temperature preference, anything like this. Okay, and it reaches a general host habitat. Okay, just consider that there is a big field. Okay, an insect flows into the wind wind takes the insect to this whole field. Okay, just consider this is a moth, any moth you can consider. Just take the example of Rusi Parda only. And you can just see in this area it is maize. Here it is some other crop. Here it is some other crop. Here it is some other crop. So the insect is entering in this whole cropping zone, big cropping zone, by using the general phenomena like anemotaxis, phototaxis, geotaxis, whatever is preferable. Like it may happen that this temperature in this zone is more suitable towards for this pest, so it gets oriented towards this. Humidity, it gets attracted towards the humidity. Anything will be the dragging factor for that insect. Okay, and once the insect reaches a general habitat, okay, after it, its sensorial, long range sensorial mechanism gets activated. Okay, long range sensorial mechanism, it uses its long range sensorial mechanism, and those are visual or olfactory. Visual or olfactory receptors it will use. It may either identify the maize plants, or either it may receive some olfactory signals from the maize plants, some volatile signals from the maize plants, and gets withdrawn towards the our maize field. Then this is the host finding. It found its host among the whole generalist field. It identified its host. Okay. Then after identifying the maize zone or host zone, Frusiperda moth entered it, and now it has to recognize it. It was getting the volatile signals from this field, so it moved to it, oriented towards it. Okay, now it has to land upon the, it has to recognize its particular host, where like you everywhere where it's not maize now, there may be weeds too in the field. So it has to recognize where its main host is thriving. So this is facilitated by specific volatiles. Okay, this is facilitated by specific volatiles and gustatory inputs. Okay, it is characteristically triggered by gustatory inputs and olfactory inputs. Okay, and when it identifies its main host, then it goes for the oviposition. It goes for the oviposition. The next step is host acceptance. Okay, after recognizing maize plant, after recognizing maize plant, it will try to distinguish that whether it can accept it or not, whether it is favorable for it or not. So if insect is of biting type, then it will go for test biting. Okay, if it will like the if the that host will pass the test of biting, if insect likes it in first bite, it will go on feeding. Okay, and if it doesn't like, it will cease. It will cease without continuing. Then the host is rejected. But if it likes the taste, the host is accepted and the feeding process is continued, and cessation will only occur when the pest is totally satisfied. Okay, this is the host acceptance. Next is host suitability. Once insect eats the plant, once insect eats the plant, this host suitability is mostly governed by the nutritional values. Okay, it is governed by nutritional values. The pest fed upon the plant. It is already after the host acceptance. It has done whatever it wants to do with that crop. Okay, it would have damaged the crop by feeding. Then the response will come. The insect has consumed the plant, and the that plant will show its nutritional effect upon the insect body. If there are some toxic compounds which insect has could not have identified in the above four processes, then those toxic compounds will go inside the insect body and will harm the insect, therefore making the host plant unsuitable for the insect. But if it is not having any toxic compound and it is having the nutritional values which enhances the insect's growth and reproduction, then that plant will become a very suitable host for the insect multiplication, and the insect population will start growing in the field. This is the whole host plant selection process. Next is the classification of resistance. This is very much important topic. Everyone should know about this thing. And here a lot of terms will come. And we have to all to deal with these terms one by one. The first basis is the based on evolutionary concept. The first basis is the based on evolutionary concept. Resistance is classified into two types. The first one is sympatric resistance and the other one is the allopathic resistance. Okay, just let's understand what these two terms means. See, sympatric resistance. In sympatric resistance, the sim matlab similar. Patric means paternal or ancestry Okay, sympatric, similar, paternal or similar ancestral history. Okay, in sympatric resistance, resistance is developed due to, see, if plant and insect are having co-evolutionary history, and due to this, the resistance is developed. Plant has developed the resistance towards insect, only then it is sympatric resistance. Okay, in sympatric resistance, insect and plant share continuous co-evolutionary history. And here resistance is evolved. Resistance is a result of gene for gene interaction. Gene for gene interaction during that co-evolution process. Okay, during that co-evolutionary process, there is a gene for gene kind of interaction between pest and the plant, and therefore resistance gets developed. And it occurs due to sympatric resistance occurs due to natural selection for resistance in plant. Natural selection for resistance in plant. Okay. And sympatric resistance is mostly governed by major genes. I will be explaining what does this major genes means in the next incoming lecture. In this lecture only. Later we will discuss when we will come to the vertical and horizontal resistance, then I will explain you what does the major genes means. Then next is the allopathic resistance. Okay, in allopathic, as you saw that in sympatric there was a co-evolutionary history between plant and insect, but in allopathic resistance there is no co-evolutionary history between plant and insect. Okay, it is just opposite of the sympatric resistance. No co-evolutionary history is present between plants and insects. Okay, and if there is no co-evolutionary history, then there is no gene for gene interaction. Then how the resistance is being developed or evolved? It has been evolved due to the pleiotropic effect of gene. Pleiotropic effect of gene, which are present as a result of selective forces unrelated to the pest or insect. See, in allopatric resistance, what happens? Plant develops resistance, plant develops resistance in absence of host, in absence of pest, sorry, in absence of pest. There is no insect. See, this is one rice plant, okay, and insect, any insect you can consider, usually just consider, and it always happens in, it mostly happens in, there is some insect, just consider for the example, this is rice plant, and this insect never came to this field. This insect never came, not this insect only, the whole species of this insect never came to this field. Okay, but another insect came to this field. Okay, but another insect damaged this plant or tried to feed upon this plant. What happens? This rice plant or this plant will is somewhat resistant towards this. It is having genes which confer resistance into this plant towards this insect. Okay, towards this insect. And when this plant is exposed to this insect, it is being sought that this plant only is exhibiting resistance towards this insect too. Okay, this plant is also exhibiting resistance towards this pest too. See what happened? It never encountered this pest. It never encountered this pest, but still it is showing resistance towards this pest. Slowly it developed resistance towards, it was encountering this pest. Slowly it developed resistance in this plant. This plant developed resistance towards this insect. Okay, and in due to this resistance development, it also developed resistance towards the non-visiting insect. Okay, this is only the pleiotropic effect of gene. The plant acquired resistance. There were some genes present in this plant which conferred resistance to some other pest, which was conferring resistance towards the first pest, and it is also conferring resistance towards the second pest, which never came to this field. So the resistance development occurred in absence of this pest. So for this pest, it would be allopatric resistance. It would be allopatric resistance. There is no co-evolutionary history evolved because both host and insect never interacted with each other. It is just due to the resistant effect of the another gene. Okay, and allopatric resistance is mostly governed by polygenes. Allopatric resistance is mostly governed by polygenes. Our next type is based on genes regulating resistance. Okay, means how many genes are regulating the resistance in the plant. The first one is the monogenic, second one is oligogenic, and third one is the polygenic. Monogenic, as name suggests, mono means one, means it is governed by single gene. It is governed by single gene. Oligogenic, oligo means few, matlab oligogenic resistance will be governed by few genes. And in polygenic, a lot of genes will be governing the resistance, will be imparting a portion of, the effect will combine together, the effect of many genes will combine together for imparting the resistance. Okay. Now there is one more thing. This based on the type of regulating gene. The another one is based on type of the regulating gene. This is the vertical resistance and the horizontal resistance. Okay, vertical resistance and the horizontal resistance. The vertical resistance is governed by one or few genes. One or few genes. means vertical resistant is mostly monogenic and oligogenic in nature. And it is specific. It is specific to. Vertical resistance is specific in nature. Whereas horizontal resistance is polygenic in nature, means it is governed by many minor genes. Okay, in vertical resistance, major genes are involved, and these are specific in nature, and monogenic and oligogenic resistance comes under this category. In horizontal resistance, many minor genes are governing this kind of resistance. And see, this vertical resistance can easily be broken. Okay, vertical resistance can easily be broken, but horizontal resistance is hard to break. Horizontal resistance is hard to break. Just consider an example like this is the one log. Okay, and one person can come and break it. But if there will be a bunch of logs, one person will not be able to break it. The same thing applies in the case of vertical and horizontal resistance. Okay, it is governed by single or few genes, then it can easily be broken. While in horizontal genes, many genes are contributing their effect, so it cannot be easily broken. It's very hard to find the pinpoint of that for that insect. Okay, there is one more thing, one more basis, that is based on multitrophic interaction. Based on multitrophic interaction, it is very much important. And one more point I forgot to discuss here in vertical and horizontal resistance. In what? Yes, say.
Ma'am, what are the major genes and minor genes, ma'am?
See, major genes, this whole classification is only just think this thing, this major gene and minor gene, what you can say, uh, as I said, now monogenic is also associated with major gene, and polygenic is associated with minor genes. Okay, vertical resistance is associated with major gene, and horizontal resistance is associated with minor genes. See, there is one gene. Okay, one gene, there is an insect. This insect is having a single gene which is imparting the resistance. Okay, single gene imparting the resistance. Then this gene is the major gene. It is known as major gene because it is the major factor for imparting the resistance. It is the only factor for imparting the resistance. That's why it is the major gene. Okay, but in minor gene, what happens that a lot of genes, this is the insect body, it is having a lot of genes. Okay, a lot of genes, like 4 to 5 genes, they will contribute, it will show its effect 1, effect 2, effect 3, effect 4. Okay, and these all 4 effects will combinedly provide the resistance. So is there any major, can you say that this gene is majorly responsible for imparting resistance? No, you will not be able to pinpoint a particular gene for imparting the responsible for imparting the resistance. That's why these genes will be considered as minor gene. Okay, they are contributing small portions for conferring the resistance. That's why these genes are known as minor genes. And it will be very much hard to break. Like if one insect is able to overcome this gene, okay, the resistant effect of this gene, in some manner it got evaluated and it counteracted this gene, but other 3 genes are still present for conferring the resistance. So even though a small part of resistance is weakened, still there are three portions remaining for defending that crop. So it is very much hard to break. This kind of resistance is very much hard to break. Is it clear now? Major and minor concept?
Yes, ma'am.
Okay. And our next, what I was dealing with the last one, the based on multi-trophic interaction. Okay. In multi-trophic interaction, there are two types, intrinsic and extrinsic. In multi-trophic, see, what does the word multi-trophic means? You all have studied the trophic levels in food chain, trophic levels in food chain. In 11th, 12th, or even lower classes also, we read trophic levels in food chain and food web. Okay. What happens there? There are some levels like primary, then secondary, tertiary, and this kind of levels we will see. Okay. In the same way, you would have studied tritrophic interaction in ecology portion. Tritrophic interaction, what does it means? That in entomology or in any field, there are three kind of interactions available. The basic one is the plant. The basic trophic level is occupied by the primary trophic level is occupied by plants. Upon this plant, who eats? Herbivores. Okay, herbivores means our insects. Okay, these herbivores are this is the producer, this is primary consumer, then comes our natural enemies. They will come secondary consumer, means first trophic level, second trophic level, and third trophic level. Okay, this natural enemy is on third trophic level, herbivores are on second trophic level, and plants is on first trophic level. Okay, so based on this, the interaction involved in the trophic level, between the trophic level, and on what basis this resistance is being imparted, these two categories are there. See, this is plant, this is herbivore, means insect, and this is natural enemies. Okay, there are two mechanisms involved. If plant directly shows resistance towards herbivores, means insect is not able to feed upon that plant itself. Okay, so what factors will be regulating? Intrinsic factor, means those factors which are intrinsic to the plants will be regulating this feeding. Just for an example, you can say uh, presence of wax layers or presence of a lot of trichomes. Okay, presence of a lot of trichomes over the leaf surface, then insect will not be able to eat the plant leaf, or it will not be able to move very frequently upon the plant leaf, and its survival and growth will be affected. So these factors are the intrinsic factors. That's why it is known as intrinsic resistance. Okay. uh, it may also be by producing toxic chemicals like allelochemicals, okay, antifeedants, repellents, and many things. Means plant is directly affecting the insect. Okay. The next mechanism of resistance is that plant is suitable for the natural enemies of that pest. Plant doesn't interact with insect. Plant is not harming the insect. Plant is not doing anything with the insect. Instead, it is creating conducive environment for the natural enemies. It is creating conducive environment for the natural enemies. Natural enemy flourishes well into that plant canopy, and thus indirectly it feeds upon that present insect. It will not, it will, natural enemy will directly harm the herbivores. So plant has not to directly interact with the insect. Plant is interacting with the natural enemy, means higher trophic level. Okay, then it is extrinsic resistance. It is means The control factor is coming from the outside of the environment. What is the control factor? Natural enemy. Plant is only providing the conducive environment for the extrinsic factor. Okay, it is bringing another factor in between. Okay, and that is indirectly controlling the pest. That's why it is known as extrinsic resistance. Okay, here you are seeing one more term, adult resistance. Adult resistance. What does it mean? There are some synonym terms towards it. Adult resistance is also known as mature resistance or field resistance, and it means cumulative effect. It refers to the phenomena where minor genes, okay, the cumulative effect of minor genes is expressed when plants are mature. Okay, mature plants are having a lot of minor genes, and when in old stage they show the cumulative effect, then it is known as adult resistance or mature resistance or field resistance. Means it usually prevents the crop from getting infected into the reproductive stage, because usually the mature resistant or adult resistance is mostly activated in the at the time of reproductive phase, and it prevents our reproductive phase, which is most crucial for the yield. Okay, this is all about this slide. Classification of resistance.
Ma'am, could you explain once more about the adult resistance?
Okay, I will. See, in adult resistance, what happens that a plant, okay, a plant is having two phases, vegetative phase and reproductive phase. Okay, vegetative phase is the early life. Okay, early life of the plant. Reproductive phase is the late life of the plant. Okay, reaching here, the plant is totally converted into the adult. The plant is totally converted into the adult, and it is nearing the maturity phase. Okay, it is moving towards the maturity phase. Then what happens that it is very much crucial to save the plant at the reproductive stage to avoid the economic losses, that at this stage no insect should infest or no severe infestation of the pest must happen. So some plants have some minor genes. I already told you, minor genes. Okay, what they do, they show their cumulative effect. Minor genes always show their cumulative effect. Okay, when this cumulative effect of minor genes present in plant is expressed at adult stage or at the later stage of plant growth, then this phenomena is known as adult resistance. It helps in protection of crop at the reproductive stage of the crop. Okay, is it clear now?
Yes, ma'am.
Okay, then we will move forward. The other thing is, and just remember the synonyms of that adult resistance. It is also known as mature resistance or field resistance. See, mature and adult resistance, you will not get confused. But with the term field resistance, you will get confused. So always remember that field resistance is synonymous to adult resistance or mature resistance. Then the next thing is we will be dealing with ecological resistance. Okay. Ecological resistance. Ecological resistance is also known as pseudo resistance. Pseudo resistance. Ecological resistance is also known as pseudo resistance. This thing you have to bear in mind. It is very important thing. Okay. What does it mean? It is apparent resistance in potentially susceptible host plants. It is apparent resistance in susceptible host plants due to conferred due to environmental conditions. Conferred due to environmental conditions. Pseudo resistance, it is apparent resistance in potential susceptible host plants due to environmental conditions. Okay. It is not the true resistance. It is not the true resistance. It is mostly conferred by the environmental condition. And it is non-heritable in nature. Non-heritable in nature because there is no genetic involvement. There is no involvement of genetic structure. There is no change in the genetic structure, or there's no difference in the genetic level. It's just like the thing, as I already gave you example, that cold susceptibility, if it resides in a hot climate, then it will obviously show the resistance. There will not be any symptom of cold. Okay. The key player is the environment here in imparting the resistance. So there are three mechanisms of this pseudo resistance. Okay, there are three mechanisms of this pseudo resistance. The first one is host evasion, second one is induced resistance, and the third one is the escape. The first one is the host evasion, second one is induced resistance, and the third one is the escape. It is very much important. Let's deal one by one. Host evasion. What does host evasion means? It is passing most susceptible stage of the crop quickly. Okay, passing most susceptible stage. The plant will pass its susceptible stage quickly. Okay, it is able to quickly surpass the susceptible stage when the pest number of insect is less. Its example is early maturing variety. Okay, some early maturing varieties are very much effective in managing in uh, in avoiding the pest attack, just because it passes the susceptible stage at the unfavorable environmental conditions. Okay, at the unfavorable environmental conditions. Next is induced resistance. Induced resistance. You should know the concept. These terms are very much important in respect of the concept. Okay, example is okay, but the thing that you should know the definition of host evasion. What is the concept of host evasion? They will ask you the question with that. They will just twist the language of the question, and they will put that concept in some twisted language, and they will ask which one it is. Okay, host evasion, induced resistance, escape, which mechanism it is. So you have to be very much clear in the concept that if the plant is passing the susceptible stage, is able to pass the susceptible stage quickly, then it is host evasion. Okay, then next is the induced resistance. Okay, in induced resistance, what we say, like you have been knowing this thing that good nutrient. Okay, we should maintain balanced nutrient. We should provide it's applicable in humans also. Like if our immunity is strong, okay, if our immunity is strong, like just consider the case of COVID. They were saying that keep taking vitamin C and zinc. Why they were suggesting? It will help us in boosting our immunity, and we will be less prone to the COVID infection. Okay, so in the same way, this induced resistance is there. What happens if our plant growth is very much good, if our plant is very much robust? Okay, we supplied it proper amount of nutrient, we supplied it proper amount of water, then the resist our plant growth will be, our plant vigor will be enhanced, and it will be less prone to the insect. Okay, so what happened in this case, that resistance was induced temporarily, resistance was induced temporarily by changed condition of plants or environment. Okay, resistance was induced temporarily by changed conditions of plants or environment. Just for example, there is very much a common thing that K and phosphorus. Okay, potassium and phosphorus are known for inducing resistance in plants. Resistance in plants. Phosphorus and potassium are known for inducing resistance in plants. So they will say that go for the balanced application of nutrients. Okay. There is one more thing. Sometimes water stress is also able to regulate the insect population. Okay. And induced resistance is mostly, it mostly occurs due to changed level of, it occurs due to changed level of tannins and phenolics. Okay, these are secondary substances. These are secondary plant metabolites, secondary plant metabolites, tannins and phenolic compounds. Okay, and with this tannins and phenolic compounds, you should know one very important bit, that these secondary plant metabolites are produced by through through which pathway, through which pathway. Okay, this pathway name is shikimic pathway, shikimic acid pathway. Okay, the tannins and phenols are produced through this acid pathway. These are secondary plant metabolites, and due to this, the level changed level of this these secondary plant metabolites, the resistance gets induced or resistance gets lost. Okay, this is also one factor. Then the another one is the escape. Escape means absence of infestation or injury to host plant. Absence of infestation or injury to host plant because of transitory circumstances. Okay, because of transitory circumstances such as incomplete infestation. Okay, just one or few individuals, one or few individuals gets affected, doesn't not affected. Just consider this is a big field. Okay, and here JS335 variety is being sown. It is soybean variety. Okay, it is very much susceptible towards as litura. It is very much susceptible towards as litura. Okay, this plant is there, this plant, this plant, this plant. There are a lot of plants in this whole field. Okay, what happens that this plant gets infected with the litura, this plant got infected, this plant, this plant, this plant, these all plants got infected with the litura. But there were some plants in field which escaped the damage of the aslitura due to any reason, due to any reason or due to any circumstances. These were able to escape the infestation of the aslitura. Okay, then it is known as escape. This phenomena will be known as escape. See, now you would be saying, many people get confused between these two terms like host evasion and escape. What is the difference? Like both will, when you will, when I am explaining, you both will be getting the same concept. Okay, in host evasion, what was happening? that insect was able, just consider this, this is a plot and there are plants. Okay, and in this, the plants were able, in host evasion, plants were able to pass the most susceptible stage very quickly. Okay, these were able to pass the most susceptible stage very quickly. So they are escaping only, in one sense they are escaping the damage. Okay, and in this escape also it is happening. But what happens, host evasion refers to the whole population. Host evasion refers to the whole population. It will not happen into a single one or two plant in the whole field. The whole plants, which whatever are present in this whole field, they will show the host evasion. Okay, host evasion is associated with the whole population, but escape, majority of the plants present in the field will be infected. Only one or two plants, only one or two plants only one or two plants will be able to escape the damage. Okay, this is the difference between host evasion and escape. So here we reached. Our next slide is mechanism of resistance. Mechanism of resistance is itself a very big topic, and I think class is already very much long. I just want you to note down some bits which are very much important for the examination point of view. The first bit is the father of host plant resistance. Father of host plant resistance is R.H. Painter. R.H. Painter. See what I'm saying, the historical perspective which I discussed in the initiation of class, there I discussed two varieties. One was the Underhill variety of wheat and one was Magetin, apple variety, winter Magetin. Okay, those two examples are very important in host plant resistance. Then what's important next? All those terminology concepts are very much important. A lot of questions are being framed from the those concepts, and they will just twist the language of the question, language of the concept, and they will ask you to choose the correct answer for that concept. Okay, and the next one is the father of host plant resistance is R.H. Painter. Then another bit you need to remember is the word non-preference was given by Painter. The word non-preference was given by Painter, and word antizenosis was given by Kogan and Ortman. Kogan and Ortman. Antizenosis given by Kogan and Ortman, and non-preference was used by Painter. Okay, this thing also you have to remember. There are some theories which you have to learn. It is very much important, like who gave which theory. Sometimes it comes in the matching sections too, and sometimes it is directly asked in the objective questions, like this theory is given by or proposed by. The first theory is the botanical instinct theory. Botanical instinct theory given by Bruce, 1920. Botanical instinct theory, Bruce, 1920. Okay, next is chemical basis of host plant selection. Chemical basis of host plant selection proposed by Kogan, 1977. These theories are very much important. You have to remember it. Okay, there is no way you can skip it. Chemical basis of host plant selection proposed by Kogan. The next is negative stimuli theory. Negative stimuli theory given by Zermi. 1961. Negative stimuli theory given by Zermi, 1961. Okay. Chemical basis of host plant selection proposed by Kogan, 1977. Negative stimuli theory was given by Zermi, 1961. The next is theory of chemosensory basis. Chemosensory basis of host selection. Proposed by Dethier. Theory of chemosensory basis of host selection proposed by Dethier. The next theory is token stimuli theory. Token stimuli theory. It was proposed by Frankel. Token stimuli theory proposed by Frankel. Theory of chemosensory basis of host selection proposed by Dethier. The next is nutrient imbalance theory. Nutrient imbalance theory proposed by House. Nutrient imbalance theory proposed by House. The next is dual discrimination theory. This is very much important theory proposed by Kennedy, Beckott and Beckott. Dual discrimination theory proposed by Kennedy and Beckott. Okay. The next is non-preference and anti. Yes.
Kennedy and
Beckott. I am writing it for you. Kennedy and Beckott.
Thank you, ma'am.
Okay. The next theory is non-preference and antibiosis theory. Next theory is non-preference and antibiosis theory. It was proposed by Painter. Non-preference and antibiosis theory proposed by Painter. These theories will be your homework for today. These things you have to learn very well. Okay. And try to do it today only, because if at once these things you will try to mug up or try to memorize, these things will not retain in your mind. So proceed with little matter, with small portions of the matter. Okay. So today your work is this thing only, that you have to remember the theory proposed by whom. Okay. And revise the concepts, whatever I taught you today. So let's conclude today's class. Hope you would have enjoyed this. And we will proceed with the mechanism of host plant resistance in the next class. And I will be giving you some tables for remembering. And there, if possible, I will try to cover the biorationals too in the next class, because there is some overlapping material which has to be discussed in the host plant resistance also and biorational control too. So there are, and even whenever you will solve the topic, or you will go for the topic wise questions, then it will cover both topics together. It will have questions of those topics. So I will first teach you, and then I will give you the questions, so that you will be able to analyze yourself. Okay. So for now, let's conclude it. Keep learning, and bye.
Thank you, ma'am.